Rumi, just let me know when you want to get started, and then yeah.
Oh yeah, I just wanted to say, do you want to get started now?
Yeah, I don't mind. Let me just see if I can share
Uh,
my screen still.
well, welcome everyone, and I hope you enjoyed this session. Also, you have to stay until the end for the link to the feedback form if you want to get a certificate of attendance. So enjoy.
All right. Let me just make sure I can still get this thing up. So, good evening, everyone. I'm Alhasan. I will be starting at Oxford next week to do medicine, but that's not very relevant for this because this is just be an A-level biology kind of revision session for Year 12 stuff. So today I've been told to go over mass transport and animals. So yeah, hopefully like we'll go through it and then some practice questions and some kind of quizzes and stuff just to get up to it. And then possibly at the end, depending on how much time we have left and any interest, hopefully it should only be really year 13 here, possible retake students as well. We can also go through some things for the AQA biology essay because I scored quite well on that this year. I will try to keep an eye on the chat. Thank you very much, whoever said congratulations. Um, I will try to keep an eye on the chat. However, I will keep asking as we go through. Oh, does anyone have any questions? Or what do you think about this? So on. So try not to bombard me. I will get overwhelmed and start panicking. I'm kidding, but yeah, so that's all good. So, for A-level biology, as I just mentioned, I always say there's like kind of three steps to tackling it. So understanding. So what I'm gonna do, I'll go through all the content for the spec point about mass transporting animals for AQA. Make sure you guys understand it because if you don't understand it, there's no point memorizing stuff. I think for the sciences, it's so important to not just go on Anki and spam like memorizing facts. If you can't actually go to someone, a layperson, and just explain what you're actually talking about and what you've been memorizing. However, this is what we're not going to be doing during this section. I think it'd be quite boring if I just shared my screen and went through Anki. So when we do memorization, but we can definitely discuss how you do that if any of you'd like. And application, that's going through your exam questions and stuff after. So I will see if I can click the slide on. There we go. Oh, that's just went send it back. Okay, so this is the spec point for mass transport animals. So we've got hemoglobin, loads and loads of stuff about hemoglobin, starting with just the structure of it and how it works, and then how it's affected by things in the animal's environment. And then it goes on to blood circulation with a little bit about tissue fluid at the end. And I understand why people want to go through this as questions on the oxygen binding and especially tissue fluid as well. Those application questions can be quite hard, and we will go through some of them and discuss them as well. But again, like I said, it's important that you understand how they work, so then you can apply your knowledge to very unfamiliar situations. Okay, so hemoglobin. So this is the first point about hemoglobin. So is they're a group of chemically similar molecules. They are all proteins, which are a polymer. And what's the monomer of proteins? Just put it into the chat. Amino acids. Someone screaming at me in capital letters. Brilliant. Amino acids. Thank you, guys. So no need to apologize. I love the enthusiasm. So amino acids are made from amino acids, polypeptides, and so on. And hemoglobin is a protein with a quaternary structure. So, would anyone like to put in the chat what a quaternary structure means? This could be a 1 mark question very easily. More than one polypeptide chain. So, it's how more than one polypeptide chain is assembled. So, for AQA, you don't need to know it in this much detail. However, nice knowledge for the essay. There's 2 alpha chains and there's 2 beta chains. So, in total, there's 4 chains with 4 of these prosthetic heme groups, which each have an Fe2 plus ion. And this ion is what binds to the oxygen and then forms oxyhemoglobin, which is how oxygen's carried around the body. So, is that all good so far? Any questions about that? Thumbs up flying through. All good, all good. No one's saying anything, so I will assume I can move on. Okay. So this is the harder part now. So hemoglobin and red blood cells transport oxygen. So the important thing for this, I think, is your keywords. So at your exchange surface of the animal, in most mammals, it'll be your alveoli. Um, Oxygen loads onto hemoglobin, and then it unloads at um, respiratory tissues. So you can also say load and unload is a pretty common one. But however, they also do quite like associate and dissociate with your hemoglobin. And hopefully, you should know from GCSE where oxygen goes and how it travels through the body. However, it comes under circulation as well. Okay, now specifically, you need to understand the oxyhemoglobin dissociation curve. Does anyone remember how we describe the shape of this curve? What we call it, the shape? Sigmoid, yes. It's basically a fancy sciencey way of saying it's shaped like an S. So, um, what we can see here is it's quite shallow at the start and the end. And it's quite steep in the middle. And the reason for this is your your first oxygen is hardest to bind. For clarification, 4 oxygens bind to a hemoglobin because each one binds to one polypeptide. So the first oxygen binds, this is the hardest one. However, when it does bind, this will induce a change in the tertiary structure of the protein, which reveals additional binding sites, so additional heme groups. So this makes the second and third oxygen easiest to bind to a heme group and associate with the hemoglobin molecule. Now, the fourth one, however, is quite hard for it to associate with it, as now the hemoglobin molecule is quite full of oxygen. So that's why it becomes shallower at the end. But the important one is the conformational change, which makes the second and the third easiest to bind. And that gives you the shape of the curve. Does that all make sense? Any questions on that? Brilliant, brilliant. Okay, so now we need to discuss something called the Bohr shift. But before we do that, we'll quickly just have a look at the graph. Sorry, I forgot about that. So just make sure you understand what the y and the x-axis are actually telling you. So partial pressure of oxygen, you might also see it written as just PO2. That's basically how we say the concentration of oxygen in the blood or the environment that we're discussing. And then the percentage saturation of hemoglobin with oxygen, not the blood, that's wrong, just the environment you're discussing. A percentage saturation of hemoglobin with oxygen, what percentage is associated with oxygen? Okay, so then we discuss the Bohr shift. And this is why at your respiring tissues, oxygen comes away from hemoglobin, moves out the red blood cell, and moves into your tissues. So the Bohr shift. As we increase carbon dioxide, I'm hoping a lot of you also do chemistry, but if you don't, it's more than fine. This might be a bit too much detail. You don't need to know this. I just find it more helpful to understand. But to clarify again, you don't need to know this. However, you can mention it in following essay stuff. Carbon dioxide, when it's in the blood, when it dissolves, it reacts with water. And it forms something called carbonic acid with like a H plus and a CO3 minus, I think. That's not balanced. I've forgotten what the exact equation actually was. It's been a while. It's been a very long time. But the important thing is you need to understand H plus ions are produced. And hopefully you should know this from topic one, your biological molecules, especially the inorganic ions bit. When we increase the concentration of H plus ions, what do we get? What changes in the environment? More acidic. Brilliant. Acidity. pH. Brilliant. So the main point is when we increase CO2, we get more carbonic acid in the blood and decreased pH. So, as there's more acid, we've got a decreased pH. What do we think pH is going to affect? Perhaps thinking about enzymes in this as well, which is also a protein. Tertiary structure. Brilliant. So, when the pH changes, it'll change the tertiary structure of the hemoglobin, and it will have what we call a reduced affinity for oxygen. The bonds in the tertiary structure. I'll extend it a little bit. What bonds exactly do you think it would affect if it's ions? The H plus ions? Ionic, brilliant. So reduced affinity for oxygen. So less association. Does that make sense? If anyone would like me to go through that again or have any specific questions, please do say. We all good? All good. Brilliant. Okay. So this is when we look at this graph. So as we increase CO2, the hemoglobin will become less saturated with carbon dioxide. So do we think it will shift to the right or shift to the left? If there's more carbon dioxide. Okay, someone said left. Quite a few people saying left. Okay, so unfortunately, I'm going to double check this because I always get this the wrong way around, but I am 99% sure it would be to the right because at the same, someone feel free to fact check me on this. At the same PO2, the hemoglobin saturation with oxygen is going to be lower. So ball shift is to the right. Someone just said. So at the same PO2, we get a lower saturation of hemoglobin, which is what we'd want. We want less oxygen to be in the blood. We want more of it. I mean, less oxygen in the hemoglobin. We want more of it in the blood, so we can go into tissues. But I please someone do fact check me on that because that's how I understand it and that's how I remember it. But again, I've been out of the loop on it for a while. So yeah, does that make sense? I'm going to quickly fact check it myself, actually. Yes, it will shift to the right. Thank you. So when CO2 increases, the graph shifts to the right. But the important thing is, I think, is when you understand biology from first principles, you can work it out yourself on the spot. So thinking, okay, there's more CO2, it's getting more acidic. It would change the tertiary structure. More oxygen leaves it, and then the percentage saturation of hemoglobin is lower. Yes, it's because we have reduced affinity. Does that make sense? Sorry about my mix-up. I got a bit confused there. So it can unload to respiring tissues. Yeah. Does that all make sense? Any questions? Could I repeat? Which part specifically? Or just all of it? All of it. Okay. So when CO2 increases, we get more carbonic acid in the blood, which means there's more H plus ions, it's more acidic. After this, when the pH decreases, because it's more acidic, the tertiary structure of the polypeptide will change because of the changes in the ionic bonds, because of the changes in H plus ions, these positive hydrogen ions. This will then mean that the affinity for oxygen is lower. So more oxygen dissociates from the hemoglobin, which then means at the same if you look at the same concentration of oxygen in the blood, there'll be less oxygen in the hemoglobin. Whereas if there's less carbon dioxide, for example, in the orange curve, there'll be more oxygen in the hemoglobin. Hannah, does that make sense? No worries. Okay, let's move on. Okay, and this is where we continue on, and this is where we have to start applying this knowledge. So, um, to different animals. So, for example, for this one, when we have a higher altitude and a lower altitude. So, I haven't done any of this topic. Do you think I should stay? I would probably advise you don't, because I'm going through at a speed that your teacher will probably go into. Your teacher will probably do it a bit slower. So I would advise you maybe don't. But again, there's a recording which you can look at another time for revision. So does anyone know the difference between the oxygen concentration in the air at a high altitude versus a lower altitude? Lower at a higher altitude. Brilliant. So the PO2 is lower here. Now, if the graph is shifted to the left, does this mean the hemoglobin has a greater or a lower altitude for oxygen? Not altitude, a greater or a lower affinity for oxygen if it's more to the left. A greater affinity. Brilliant. So we can look at that. Like if you couldn't work that out, for example, at 4 partial pressure, the saturation is higher for the one to the left, whereas at the same one, saturation is lower. So that means there's more oxygen in the ones at the ones to the left. So, why do you think at a higher altitude we'd want hemoglobin to have a higher affinity for oxygen? To easily load, not quite unload, but it'd be easier to load because the affinity is higher. So it can associate at a lower oxygen. So you want to get as much of it. That's a brilliant way of thinking about it. You want to get as much of it as possible. So you want your hemoglobin to associate with a lot of oxygen so it can deliver it to respiring tissues for a respiration. Affinity means like how much they attract to each other. So how easy it is for oxygen to bind to a molecule of hemoglobin. So if hemoglobin has a higher affinity for oxygen, oxygen is more likely to bind to hemoglobin, if that makes sense. So that's one way you can apply your knowledge. You may also be given in the example of just organs of different surface areas and just what environment they live in. And you just need to, with I think the whole like subtopic of hemoglobin, always just work back from first principles and then work out what you need to do from there. Does that make sense? Anyone wants to go through anything again? Okay. All good. Okay. So, any final question about anything to do with hemoglobin from anything I've been through? How would you answer a question on high altitude in exam questions? Like the order. So, honestly, the order doesn't really matter. But again, if you just work from first principles, so you think, okay, at high altitude, there's a lower PO2 in the partial pressure of oxygen in the environment. So, um, the hemoglobin has a higher affinity for oxygen. More oxygen will associate with hemoglobin. So, more oxygen is delivered to respiring tissues to be used in aerobic respiration to release energy. Does that make sense? So, just work through it in like a logical sequence, if you get what I mean. Brilliant. Okay. No worries. Um, so, anything else on hemoglobin, or we'll fine? I'm assuming we are. Okay. So blood circulation in a mammal. So hopefully this diagram should be, please don't write on the screen because everyone can then see it. Do you have a generic mark scheme for this? There'll be some towards the end, but I think my main advice of ill biology, which I'll discuss later, is genuinely when you get a question, write everything relevant to it. You're like, if it's a 2 mark question, you shouldn't just be writing 2 points. You should be writing 3 or 4 that you know is right. Never write stuff that's incorrect because that could lose you marks, especially to reject. But most of the time, I'd say 90% of the time, just write what you know that's related to a question because you never know where you'll pick up the marks. Um, so like if like in the future when like in like 2 years, if I ever go through any of my A-level papers or if I ever find any of my mocks, I can literally show you like sometimes for a 2 mark question I'll write 5 or 6 points just to guarantee that like I've got secured the two. And I think this is what AQA are going for. They want you to over explain and show your knowledge. So that's why the mark scheme is quite niche, so that they can pick out. So like to have explained it in the detail that they want you to explain it to hit, you need to explain it in a lot of detail to hit those points. They are very harsh, but that's how I worked around it, if that makes sense. So hopefully you know this from GCSE. The left side of the heart pumps your oxygenated blood to the rest of the body, and the pulmonary arteries take and vena cava carry your deoxygenated. So yeah, the only thing that's not mentioned here is the kidneys. Does anyone know what vein and artery carry blood away and to from the kidney? Because there'll be a recording sent us. There is a recording. Did you do A-levels in 2025? Yes. They were. I can't talk about them too much, but I had mixed feelings on them. I think, however, they were standard papers. Just people were stressed because it was their exams. Renal, though, yes. In respiring tissues, why is the partial pressure of oxygen low? So it's because oxygen's being constantly used up. I didn't see. I'm seeing. Yeah, but it's renal. So the structure of the heart is quite important to understand, and also how blood moves through the heart. So for the one thing to remember as well is the left and the right side are reverse. So left and right. Imagine there's someone like lying down in front of you, and we're absorbing from their left and their right. So for the right hand side, that's where the oxygenated blood moves through. Pulmonary artery. One piece of advice I'd give is you may see things like tricuspid, dicuspid, those not dicuspid, bicuspid. You don't need to remember them. I don't think there's much point. I would just advise semilunar valve and semilunar valve and atrioventricular valve. You don't need to remember the fancier names, and it's a lot easier, I think, to remember those, especially atrioventricular valve between the atria and the ventricles. Semilunar, you just need to remember, is the one going out of the heart. Does that all make sense? Okay, so let's talk about valves now. So what I'll do is I'll quickly zoom in on, let's say this is the right atrium. So we've got atrium, got your valve, and you've got your ventricle. So when the heart contracts, the first thing that happens is the atria contract, which is what we call atrial systole. Now, when a chamber of the heart contracts, what happens to the chamber of the heart? What increases? Pressure. Brilliant. So the pressure will increase in that chamber. So in your atrial, during atrial systole, the pressure of the atria will increase. So now the pressure of your atria, the volume decreases, the pressure increases. Brilliant. That's a really nice way of thinking about it, because if there's less volume, because if the volume of the actual chamber decreases, but the amount of liquid, the blood inside is the same, is pushing out, it can make the pressure increase. So now the pressure in the atria is higher than the ventricles. This will cause the valve to open. So the valve opens. and blood moves from the atria to the ventricles. Okay, does that make sense? Okay, so our first step, atrial systole. And then next up, what beats next? What contracts next? What bit of muscle? Your ventricles, brilliant. So we call that ventricular systole. Spot on. And this means the pressure in the ventricles will now be higher than the pressure in the atria. So this atrioventricular valve is going to snap shut. This prevents the backflow of blood. So none of that's going on. The atrioventricular valve is closed. However, now the blood pressure of the ventricle is higher than the pressure in the artery that's leaving the heart. So whether that be the aorta or your pulmonary artery. So this means a semilunar valve can open and blood will leave the heart, going via the going out of the heart via the artery. Okay, then the last step, does anyone know what it's called? So we've got atrial systole, ventricular systole, and then right, the things I don't know how to say. Is it diastole or diastole? I don't know. I know how to spell it, but I don't know how to write it. So it's diastole, diastole, diastole. Okay, I'll trust you on that. Diastole. So that's when all the muscle of the heart, all the chambers is relaxed. What did I say about ventricular systole? So the ventricles contract, pressure in the ventricles increases, which is higher than the atria. So the atrioventricular valve closes, but because it's higher than the artery, the semilunar valve opens. Systole meaning movement, yeah, or contraction. So diastole happens, and this means that the atria can fill with blood. Yes, there will be a recording. So the atria fill with blood from the veins, and that's all. Does that all make sense? Does anyone want me to go through any of that again? Diastole, I just went through it. Is when all the chambers are relaxed. So this means that all the blood just will. Your battery's going down fast. Thank you for letting me know. I'll have a look. It's probably because I'm like screen sharing, but also like to my iPad and a lot's going on. Thank you. I'll have a look at that in a second. Yeah, the renal part is year 13, how blood flows through the kidney specifically. All you need to know from AS is blood, and for your paper one, blood goes into the kidney via the renal artery, goes out via the renal vein. That's all. So yeah, diastole is when all the chambers are relaxed. So this means that atria can fill with blood, and the semilunar valve will close because the pressure in the ventricle will then be lower than in the arteries. Does that make sense? Any final questions on that? Okay, but I think we'll move on from that then. Okay, so the next part is, I think, one of the easiest parts of this topic, just the structure of arteries, arterioles, which is very, very similar to arteries, and then veins in relation to their function. So just start pointing stuff out to me. What are these layers called? So the first layer and the second layer, what do we call these? Not quite endothelium, that's the one right on the inside. That's lining it. But nice try though. Elastic tissue, brilliant. So let's say it doesn't really matter. Number two would be like probably your elastic tissue. And then number one would be muscle, brilliant, muscle tissue. Now, the important thing to understand then as well, so we've got these three parts. We've got your endothelium, elastic tissue, muscle tissue. something that's really useful to think about as well for endothelium and epithelium, because I used to always get them mixed up. I'd never know which one to write. Endothelium is blood vessels. Think if it doesn't have any contact with the outside of your body, it is not it is not epithelium. If it's epithelium, so even if it's like in your lungs, if like technically you could get like a piece of string and like wind it down the right way, or like a piece of like metal wire, a bit gruesome, but you can wind it the right way and you can touch it. It's without piercing anything. It is epithelium because it's coating the outside. So that's your digestive system is epithelial tissue. Your blood is endothelial. Your kidneys, that would be epithelial mostly, um, because that is like if you threaded it the right way, it would go. You could like touch your kidney tissue. So things like that. That's something to bear in mind. But for blood vessels, it's endothelium tissue, and especially that's important as well for when you're discussing alveoli and capillaries. Capillaries endothelial, alveoli epithelial cells. Okay, so endothelium, elastic tissue, and muscle tissue. And the important thing to understand is what their purpose is in the artery. So, muscle tissue. What does muscle do? Contracts. Brilliant. Contracts and relaxes. So, why is this contraction and relax and contracting and relaxing important? Controls blood flow. Okay, good start. I want more detail though. Pressure. Okay, so pressure. Controlling the pressure of the blood flow. So we want to maintain the pressure of blood flow. That's the main purpose of them. So they can contract and relax to maintain blood pressure. Because obviously we want blood to be leaving the heart at a very high pressure. So when it goes down to, for example, all the way to the other side of your body, going to your toes, or going, for example, like through loads of blood vessels through your brain, we want it to be at a high pressure so it can go to those extremities. So maintain blood pressure, brilliant. Elastic tissue. Thinking about the pressure again, why do we need elastic tissue in the arteries? More of it than in a vein. Recoil. So it can stretch. That's the important thing to say stretch and recoil. So maintain smooth blood flow, kind of. So it can stretch without damage, kind of. So the important thing is, what do we want to withstand? What was I just discussing? High blood pressure. Spot on. Very nice. Okay, high blood pressure. So endothelium. There's something about it which is quite important. What is special about the endothelium? Does anyone know? That especially like the surface of it is smooth. Brilliant. So it's smooth. What's important about it if it's smooth? Reduces friction. Brilliant. So this makes it easier for blood to flow through the arteries and doesn't slow it down. So as it's smooth, reduces friction. And something else I was thinking of. Someone said no valves. And the thing is with valves and arteries, sometimes, and a question that's tricked a lot of people out in the past was one about the aorta. And it was like describe the structure. And there was a marking point which a lot of people missed. Because when we think back to that last slide about the aorta and the heart, some arteries do technically have a blood valve, a semilunar valve. So that's something to be careful of. Sometimes for the aorta, and you can say, oh, some arteries do have valves, but commonly they don't. So if you get a question about the aorta, for example, the pulmonary artery, you can then go on to mention the semilunar valve and why that's important to prevent backflow. But we'll talk about valves more in a second. Okay, so some of these do apply as well to the veins. So smooth endothelium, I can mention that for that as well. And the main thing here is we've got thinner layers of tissue because blood is traveling at a lower pressure. That's just the main thing you need to know. And that they have valves. Veins have a lot of valves. Why do they need the valves? Prevent backflow of blood. Brilliant. So, yeah, is that all fine? Any questions on that? Brilliant. We'll keep it moving then. Okay. So now let's discuss capillaries and tissue fluid. So capillaries and capillary beds are very important. Like in your body, there's loads of capillaries, like just in a layer, and they exchange loads of different nutrients and oxygen and carbon dioxide with your tissues, um, glucose, amino acids, so on, so forth. I'm sure you guys know that. But let's look at how the capillaries are adapted for that. So, what is important about them, about their endothelium this time? It is one cell thick. Brilliant. Now, with that point about one cell thick, you want to be so careful when you're discussing the alveoli. Another common mistake that people make in exam questions, but prevent proteins, we'll get onto that in a second. Because they're one cell thick, but the alveolar walls are also one cell thick. When you're discussing it, you want to be so careful not to say, "Oh, the diffusion pathway is one cell thick." So yeah, I kind of just mentioned the word just then. Why is it great that they are one cell thick? Shortest diffusion pathway. Nice, 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 nice. Rapid, yeah, reach scientists. So diffusion's faster. This is a cross section of a capillary. Okay, that's the main thing you need to know about them. That they form a bed of capillaries. But the main point is they're one cell thick to allow for diffusion. Okay, now we need to discuss tissue fluid formation, which is made to seem like a very, very hard topic, but honestly, again, it's hard. It's again just understanding it from first principles. All three are endothelium. Yes. So anytime you get asked about um, like blood vessels, all of them endothelium. Obviously, it's different kinds of endothelial tissue. The cells will have differences between them, but you refer to all of them as endothelium. This tissue lining, like the inside or something. So, um, what we have here is like, let's imagine this is a capillary. We have an arterial end, of a venular end, which is what we call like, okay, so this is coming from an artery, this is going into a vein. And then you have your tissues, and they are surrounded by tissue fluid. And a point for the essay, the formation of tissue fluid is really important for making sure that, because the tissues bathe in fluid and it's got quite a lot of nutrient stuff, and it's how they can then cross into cells, because obviously if things diffuse faster through fluid as well. So, and then we've also got lymphatic draining. So when blood comes through the arterial end, because arteries are bigger than capillaries, obviously there's like a bit of a gap, but arteries are bigger than capillaries. So as we go into the artery at the arterial end, what's going to be higher at the arterial end than at the venular end? Hydrostatic pressure. Brilliant. So in general, higher pressure. But what hydrostatic pressure means is just the pressure of the water. Oncological pressure is not something you need to know for AQA. It's not a term that they use. So you just need to remember hydrostatic pressure and water potential, which we'll get onto. So as there's a higher hydrostatic pressure, water is forced out between what we call fenestrations in the endothelium. Yeah, that's the thing. Like a lot of teachers teach stuff outside of the spec as well, but if you do know it, it's great, but also you won't be asked about specifically, and it normally isn't in mark schemes either. So in these fenestrations in the capillaries, like the gaps between the cells, water is small enough that it is forced out at the capillary end. I think I've only seen osmotic effect in mark schemes. I've not. I've only really. We'll get on to osmosis in a second. At the arterial end, it's just being forced out. being forced out of what it is forced out of the capillary via the fenestrations between endothelial cells. So as water moves out at the arterial end, there's a lot more water here. So how will this affect the water pressure, the water potential of the tissue fluid? I'll grab a charger. What will happen to the water potential of the tissue fluid as yeah, so the water potential is going to increase. So the water potential will now be higher than in the blood. So what will water do? If the water potential is higher in the tissue fluid blood, move out via osmosis, via the potential, it'll move back by osmosis. So what you need to remember, arterial end, water just forced out due to higher hydrostatic pressure. At the venular end, osmosis. However, any excess tissue fluid is just drained by the lymphatic system and then returned to the blood. Does that make sense? Does anyone want me to go through bits of that again? I'll wait a few seconds because this is. Are we all fine with that? What does the lymphatic system do? So it's basically a network of just so all your tissues. Tissue fluid is incredibly important because it bathes all your tissues. If you ever think, what are my organs just sitting in? Are they just sit there? Don't we need to talk about proteins? Kind of. What you need to think with the capillaries, the reason why. Can you summarize it from arterial end? Okay, so talk with proteins and things in the capillaries. You talk about those because they lower the water potential of the capillaries because they can't be forced out. So that's the main thing with those. Okay, can you summarize it from arterial end to increase in water? So at the arterial end, as it's coming from an artery, which has like quite high pressure, the hydrostatic pressure, which is the pressure of the water, is higher. Yeah, there's a lower hydrostatic pressure. So as there's a high hydrostatic pressure, but in, for example, in the capillary, you've got these gaps between the cells, but because they're quite narrow, only water can really be forced out. So water's forced out into the tissue fluid, so there's more water, and then an increase in the water potential. Does that make sense? Yeah, brilliant. Okay. Any more questions? I think someone's asked me again about the lymphatic system. So just some extra information about that. If you ever think, oh, what do my organs just sit in? It's tissue fluid. That's what they sit in. So quite a broad term describing lots of different fluids that your organs might sit in. They have roles within your immune system. A lot of immune cells move through your lymphatic system as well, looking for pathogens and also killing things like cancer cells. So your lymphatic system is quite important. There's a lot of interesting reading you can do about that. Okay, so now it's your turn to have a go at some of these questions. I'm just quickly going to do something just so I can see the speaker notes as we go through these. Oh, something the way around. Just give me 1 second. How you'll answer them, I'll give you guys like for like a 2 marker, like 2 minutes to think about it, and then just put when you're done in the chat, and then after we'll move on. Okay, so that should be fine now. I just need to move that onto the other screen. Okay. Okay, so have a go at this question and put in the chat when you've done it. Oh, not the answer, just if you've done it. Sorry for not being more clear about that. No worries, don't no need to apologize. Can I delete messages from the chat? So let me do that. No, no worries. Brilliant, thank you. So I'm guessing about three of you are done so far. Wait for like a few more to be done. Okay, so I will quickly offer the opportunity for one person to unmute and discuss this question through. If no one wants to do that, it's more than fine. But if you do put your hand up, and then I will choose someone. If you go back and explain something, it's so useful for your learning. My mic doesn't work. Someone would like to volunteer, that'd be great. If not, also more than fine. I'm a bit scared. Nothing to be scared of. No one should really be judging. Okay, Ace, if you'd like to unmute and like just start explain what you think your answer would be.
I'd say the first answer, the first oxygen will, when it binds to the heme group, it will affect the bonding in the tertiary structure of the hemoglobin. And that means that
Brilliant,
the heme groups are more exposed and the structure changes for the hemoglobin after that. And this means that the second oxygen can bind more easily to the heme group since they're more exposed now.
brilliant. So you covered both the points in the marking. Brilliant. So binding changes tertiary structure. Um, Brilliant. So binding of oxygen to heme group, nice. And you said it uncovers another heme group for oxygen to bind to. So perfect. And that bit at the end wasn't necessary. But for example, if it was like a 3-mark question or if it was an explain, that's fine. But also you did just reiterate what was in the question, which is also more than fine to make sure you're giving a clear answer. But with that, also be careful. Like if it's a 2-mark question, you're thinking, oh, I can only think of 2 points. But if you said what's in the question again, that's nothing as well. But brilliant answer. I'm happy to move on. Can't believe this process goes to my body. I know it's quite cool how intricate everything is, but like, yeah. So when an oxygen association when an oxygen dissociation curve is shifted to the left, um, explain why this happens. What's happening in the hemoglobin that is making it shift to the left? I'll set like a 2-minute timer for this one instead. But again, putting it in the chat when you're done is quite useful. It's been about a minute. And there's the timer. So only 4 of you have said you're done. Would any of you like any extra time to think about it? Or are we all right just to move on now? Okay, again, I'll offer for someone different this time to volunteer and give an answer, but if not, that's also fine. And a few people can just put theirs into the chat and discuss them. Okay, so we'll just move on then. So if anyone would like just put their answer into the chat and then we can discuss what we think it is. Okay, so I think a misconception was, I think this was a quite confusing question. So what you need to be aware of is as well, high affinity for oxygen. Okay, easily but unload less readily. Okay, so what you need to be careful for this one, so the oxidation curve is shifted to the left. Now, what this is going to mean, as a lot of you said correctly, that oxygen, there'll be a higher affinity for oxygen, which I kind of got mixed up. No, it's fine. So if it shifts to the left, there'll be a higher affinity for oxygen at lower partial pressures. So that's great. And I think someone did say there'll be that's when we start to get. Oh wait, no, it's fine. So oxygen will load more readily, so that oxygen can be delivered to areas with lower partial pressures of oxygen and for aerobic respiration activates muscles. So you don't need to discuss carbon dioxide always. Bore shift is something different to just the dissociation curve. Lots of different things can affect the dissociation curve, things like blood pressure and temperature as well. So if it doesn't mention carbon dioxide, you don't need to discuss it. So the reason it shifts to the left is hemoglobin has a lower affinity for oxygen. I mean, a higher affinity for oxygen, sorry. At lower partial pressures. And then what this means is it's easier to load oxygen to hemoglobin. And this then means that aerobic respiration, there's more oxygen available for aerobic respiration. And there's also other points you can write, like anaerobic respiration should be delayed because there's more oxygen or there's less lactate. Does that make sense? Brilliant. Okay. Um, so describe the changes in the dissociation curve for a higher metabolism organism and why they occur. Again, I'll set like maybe a 3-minute timer for this one, so everyone gets a chance to give it a good go. Okay, so a high metabolism organism will respire more. So that's something to bear in mind. So that's one of the marker points that more respiration is going to happen, or a higher rate of respiration. 1 minute to go. Okay, that's 3 minutes done. Does anyone need any more time to think about this one? Are we all good? Ready to move on? Okay, so again, anyone would like to put the answer into the chat? I will go through the mark scheme in a second. But if anyone would like to put the answer into the chat. Okay, so the answer I've been given would score. So oxygen social curve for high metabolism shifts to the right. This is such as respiration. Okay, so what we'd need is so there's a higher rate of respiration that would get the MAT scheme is the curve moves to the right due to hemoglobin having a lower affinity. However, I think this is stupid that this is the first point because like it's a lot easier to work backwards. So to think, okay, so a high metabolism organism will be doing more respiration. So what do you need for respiration? You need more oxygen. So what we then need is that's when we get hemoglobin should probably have a lower affinity, so that more unloading of oxygen from hemoglobin. Does that make sense? So those are your 4 points. More respiration, more oxygen, more unloading of oxygen from hemoglobin. So the curve moves to the right as hemoglobin will have a lower affinity. Any questions on that? Anything you want me to go back through? So the Bohr effect is only for things relating to carbon dioxide. Anything through the Bohr shift, Bohr effect, that's only when carbon dioxide starts getting involved. So no, you don't need to mention it unless carbon dioxide is directly mentioned. Okay. So next one, atrial ventricular systole. Make sure that you mention the valves that are involved and when they open and when they close. So I will set a, let's say, 4 minute time, maybe 3 minute. Let's go with a 3 minute timer for this one. Okay. About 30 seconds left. Okay, so put your answers into the chat. Okay, so I'll have a look at this one that was just put. So okay, so with this one, the main things you need to discuss with anything to do with how blood moves through the heart is pressure, because that's what changes the valves, and what contracts when. So during atrial systole, so your first point, atria contract, atria have higher pressure. Did I put 5? Let's put 6. Something that says 6. So atria contract, giving atria a higher pressure than the ventricles. Oh no, I can't. Oh no, never mind. I thought those 2 separate points. Than the ventricles. So the atrioventricular valves open. And then after that, the ventricles, when they fill with blood, but you don't even have to talk about the blood, ventricles have a higher pressure. So then the atria. So the atrioventricular valves slap shut. So then after that, the ventricles will then contract. So the ventricles have a higher pressure than arteries. So the semilunar valves open. And then for the last point, second to last one, semilunar valves close. Again, for a similar thing, the ventricle, to the second point, the aorta fills with blood, so then the pressure in the aorta or your arteries is higher than the ventricles. You don't always have to talk about volume, but I think thinking about, if it helps, thinking about in volume can help you to understand why the pressure increases sometimes. And your last point is just saying anything about how the contraction leads to an increase in pressure at any point in your answer. So that's what the 5th marking point is for. Does that make sense? Okay. Got 2 left. And then there's some proper example questions that I'm debating. So describe how the aorta is adapted for its function. Well, set timer. I'll set a 2-minute timer now. Okay, so put your answers into the chat, even if it's just 1 point from your answer, that's more than 5. I have no clue what tunica media is. Tunica media is, but it would be a lot easier to say the aorta has thick walls because that is what's in the Marta scheme. So thick walls composed of smooth muscle. You don't need to say smooth muscle. The aorta contracts, decrease times, and increase the pressure. So that's 1. Okay, so I'm not sure what example you do, but the details about the tunica, like describing the anatomy that way, um, isn't quite mentioned in AQA, and it does actually um lose you marks because it's like, so for example, the tunica intima, um, because you've got to mention like smooth endothelial wall, um, yeah, so that's something to be very careful of, um, and like cartilage again not mentioned on the spec, so that doesn't quite get you marks. Um, so yeah, I think like with like AL biology levels in general. Oh, I take OC. Okay, that makes a lot more sense. Ignore everything I just said then. I'm not sure if that's a good answer for OCR. It could be, but yeah. So I'm not sure. Sorry. Um, but for if this was an AQA question, the marking points would be um, so you've got elastic tissue to stretch and recoil, and a weirder point, elastic tissue stretches when the ventricles contract. Again, muscle for contraction. That's literally all you need to say. A thick wall, withstand pressure, and a smooth endothelium to reduce friction. And last point, semilunar valve to prevent backflow. Does that all make sense? To do with the aorta having arteries. So the aorta is an artery. Length? What do you mean length? How it's able to reach everywhere? So aortas themselves, like the aorta itself, isn't particularly long. It branches off into other arteries. But what you could think about for the aorta is they're not particularly long. It's just the pressure, which means they can get to extremities. It's more of an application question, so it needs to apply that the aorta is an artery, very slightly, but yeah, hopefully it. But also, there's like ones which are specific to the aorta, like the semilunar valve. But yeah, it's kind of an application question because you've got to link your knowledge about one the artery, that the aorta is an artery. Okay, this is the last one on these kind of like more recally ones. So just describe how the atrioventricular valve means a unidirectional flow of blood. I'll set like a minute timer for this one. Okay, so again, put your answers into the chat for this one. Okay, so you don't need to detail all the heartstrings again, but I will benefit the doubt. I'll assume that you're not doing AQA. These cells are open due to pressure behind them, pressure in the atria. So when the pressure in the atria increases, the valve opens. So what you need to do is you need to talk about, so close. But the difficult thing for this question is, okay, so the valve opens when there is a higher blood pressure in the atria compared to the ventricles. That's the important thing. You need to make sure you're comparing the ventricles to the atria. Yeah, I think I did notice that. I was thinking, but because it's in the question, I think it'll be fine. To allow blood to flow into the ventricles. Brilliant. Valve closes when there is a high blood pressure in the ventricles compared to the atria, preventing the black flow of blood. So that is a 2 out of 2 answer. That one. That's in the chat right now. Yeah. I'd be happy to wrap it up there. I've got some more like application exam questions, but they're literally just from physics and maths tutor. But like again, I always float around on one of the group chats, and a lot of you will be giving my number and stuff as well. So I'm ready to get in contact. Any final questions, anything we've been through today, even or stuff just about generally about medicine applications or A-level biology or chemistry or maths, I'd be happy to take. Tips I'm currently, any tips? What? How do you revise bio A-level? Again, just the 3 steps, making sure you understand the content, coming to things like this and making sure and having someone teach it back to you, but also things like Miss S struck. Just reading the textbook and sitting with the content and thinking through it and making sure that you understand the logical sequence behind it. And then memorizing it, so Anki, mind maps, blurting. Blurting is amazing for biology, especially these processes. What do you do for the bio essay? That could be a good session in the future. But yeah, and then loads and loads of exam questions and past papers. Bio essay, Miss Estruck's video is amazing for it. How you plan it out. Just making sure you're focusing on an even split between importance and like just describing processes. Tips for year 12, take every opportunity you can get and just focus on learning the content this year for AS. For Chem, loads of exam papers and maths, again, lots and lots of practice. How do you do well in Chem? I just did loads and loads of past papers and past questions, and it's the same as biology, sitting with the content and making sure you understand it. What resources? Past papers, past papers, past papers. And there's some good YouTube as well, like Allery Chemistry and stuff, and physics and maths tutor the notes. But I'd be able to leave it there. Do you have any tips for the UCAT? I'm guessing you're a year 12, so I'm going to say don't worry about it, because I swear today's the last day you could sit the UCAT. But you got exam questions from physics and maths tutor, has loads, but also your teacher as well. They can make like set packs of exam questions for like a more specific topic. But for the UCAT, I'd say from year 12, don't worry about it. Start worrying about it in a few months. Enjoy these first few months of year 12 there. You don't have to worry about it too